I'd like to begin with an acknowledgement of country. Based in Wollongong, I'd like to acknowledge the Darawal, Yuin, and Wadi Wadi people as the traditional custodians of the country in Illawarra. We pay our respect to elders past and present and emerging, and to all Indigenous people living within the region, and extend that respect to other Indigenous people with whom we engage. Through our research and engagement, we seek to help others appreciate and act in respect of the significance of country. Today, we're lucky enough to have Professor Megan O'Mara with us from the Australian Institute for Bioengineering and Nanotechnology at the University of Queensland. Her research aims to understand membrane lipid biochemistry and how the modulation of membrane protein fusion impacts cell surface interactions, with her work spanning across computational structural biology, membrane biophysics, and bio-inspired polymers. Megan completed her PhD at ANU in 2005 before moving to the University of Calgary in Canada, working on membrane protein structural dynamics. She returned to Australia in 2009 on a UQ postdoctoral fellowship and was awarded an ARC DECRA in 2015. She is currently Associate Editor for RSC Advances, the President of the Association of Molecular Modelers of Australasia, and was previously the Secretary of the Australian Society for Biophysics. So we've got an exciting talk coming up today.
Thanks a lot for that, David. Really appreciate it. And thank you everyone for joining in and for listening to me. I would also like to start with an acknowledgement of country. If I can get this slide. My slides aren't working. Okay, here we go. So the work that I'm going to present here was conducted while I was at the University of Queensland, and also before that at the Australian National University. And because of this, I would like to acknowledge both the Turrbal and Jagera people from the Brisbane region and also the Ngunnawal nations in the Canberra region. I pay my respect to these people, their ancestors and their descendants, and I acknowledge the people of these lands and their oral and traditional forms of knowledge. So with that, thank you for having me here. I'd like to talk about chronic pain. So chronic pain, many of us have woken up with a headache, had stubbed our toes, things like that. And we've all experienced intense acute pain from things like this. However, chronic pain is a different kettle of fish altogether. This is a pain that lasts for longer than 3 months. It affects the lifestyle and the well-being of many, many people. In fact, 1 in 5 people around the world suffer from moderate to severe chronic pain that impacts their daily life. It's got a higher socioeconomic burden than diabetes, cardiovascular disease, and cancer combined. And it is a major form of chronic pain that's induced from a myriad of different causes, including, most commonly we think of it as injury, but it can also be an inflammatory pain, and it's also associated with neurodegenerative diseases such as multiple sclerosis, Parkinson's disease, and Alzheimer's disease. So because there are so many different causes of chronic pain, it's very difficult to treat, and there's no one-size-fits-all treatment of chronic pain. So at the moment, one of the most common treatments for severe chronic pain and moderate long-term chronic pain is opioid-based drugs. Now, these are ineffective long-term. There's no really good alternative treatment options. There's been a 300% increase in prescriptions for opioids over the last few years. And there's been associated with this a 600% increase in opioid overdoses. Now, these figures are mainly from the US, but this is also something that's been noted in Australia as well. So currently in Australia, we spend $73.2 billion on chronic pain. and it's projected to blow out to 2.15, sorry, $215.6 billion in Australia by 2050, and a kind of proportionately high figure in New Zealand as well. So this is a big problem because we've got these drugs that don't really work very well. They have terrible side effects. We have increased dependencies and overdoses from them, and we've got no really viable treatment options. I know there's a few different things coming to the market, and someone asked me the other week about sodium channel blockers. I think, this is great, but overall we need to have a whole range of strategies for treating chronic pain. And today I'm going to talk about what I do as a computational biophysicist or computational chemist, and how my collaborators and I are going about treating or finding targets for neuropathic pain, chronic neuropathic pain. So as I said, I'm a computational person, so I have a simulation toolbox for biochemical processes, and you can see here that we have a different range of length scales in terms of the system sizes we use, and these can range from the picometer, which are just bond vibrations and so forth, up to a micrometer scale size, which is approaching patches of the cell. And eventually, one of the grand aims of computational simulations is to be able to simulate an entire cell in atomistic detail. Now, as well as simulation sizes, we've got timescales in here on the x-axis, and these range from femtosecond bond vibrations up to microsecond to millisecond timescales, which are motions of protein interactions in terms of conformational changes, formation of domains in lipid membranes, and so forth. So you can see here the different techniques that correspond to each timescale. And so down in this very short length scale and size scale, we have quantum mechanics. Most of my work focuses on molecular dynamics, atomistic molecular dynamics, and also coarse-grained molecular dynamics. And these are the two methods that have been used in this presentation today. So now that I've given you a bit of a background about chronic pain and the methods itself, I'd like to introduce you to glycinergic neurotransmission. So the glycinergic pathway is an inhibitory pathway for chronic pain. And so what that means is as you transmit a glycinergic signal up to the brain, then it's a signal that inhibits the pain pathway. So in the presynaptic synapse, nerve ending synapse, I hate these things. You start giving a talk and all the words fly out of your brain. Anyway, in the presynaptic terminal of the synapse, we have glycine that is released into the synapse, and then glycine goes and it binds to the glycine receptor on the postsynaptic side of the synapse. This induces the signaling of chloride ions that propagate through the nerve, and this chloride signaling inhibits the pain signal. Now, if we're to keep glycine in the synapse longer, that will, we hypothesize, increase the inhibition of the pain signal. So what we want to do is keep glycine in here. The glycine transporter in the presynaptic terminal recycles glycine to go into vesicles to be released into the synaptic cleft. Now, if we attenuate the glycine transporter, we can slow the rate of clearance of glycine from the synaptic cleft. And we hypothesize that this will allow us to modulate pain by inhibiting the pain signal for longer. So when we look at glycine, GlyT2, which is our protein in the presynaptic cleft terminal, we have conformational cycling of this protein where it adopts an outward open conformation where it is open to the synaptic cleft, and glycine combined, it then goes into a closed conformation and then an inward open conformation where it releases glycine into the cytosol to be packed into vesicles for re-release. So these are the basic conformations, and what we want to do is be able to inhibit this process here. So in order to do this, my collaborators and I have been collaborating with Rob Vandenberg from University of Sydney and his colleagues, and also with Tristan Rawlings from UTS. So Rob and Tristan came up with a series of bioactive lipids that were present, and they looked for ones that were present in the central nervous system. And of these, we've got three here: N-arachidonyl glycine, which is an inhibitory neurotransmitter that's found in the spinal cord, and it reduces pain in rodents. We also have oleol L-carnitine, which is a fatty acid that is found throughout the body and also reversibly inhibits GlyT2. We tried a third one as well, anandamide, which is also found in the central and peripheral nervous system. It's the active compound in chocolate. And while it makes you feel very good after eating chocolate, it binds to opioid receptors, and unfortunately, it does not have any effect on GlyT2, so it does not treat chronic pain. So that was our negative control. So from these, we took N-arachidonyl glycine forward to use this as the basis of a series of bioactive lipid derivatives that we could use to test to see if they were specific against GlyT2 and could be used for chronic pain or for the treatment of chronic pain. So Tristan Rawling came up with this series. He ended up with a series of almost 100 different lipids, and these were tested using electrophysiology on xenopus oocytes by Rob Vandenberg at University of Sydney. And then we came up with this series of compounds, and a few of these we took forward to see whether or not they worked in molecular dynamics simulations, and if we could use these to try and work out the mechanism for GlyT2 inhibition and to see if they, for pain inhibition. So we asked the question, how do these actually work on GlyT2? These are lipids. So they could be working indirectly by binding and partitioning into the bilayer itself, and then inducing changes within the bilayer that may affect the conformation of GlyT2. Or they could be acting directly on GlyT2 itself by binding to GlyT2 either from the solution or the bilayer. So this was done by a student, Alex Schumann-Gillett, who finished in my group a few years ago, and she found that they did not change bilayer properties. So these compounds did not affect the properties of the bilayer, and so from that we deduced that they were binding directly to GlyT2, either from the solution or from the bilayer. And from that, we ended up doing a series of molecular dynamics simulations where we looked at the spontaneous binding of these lipid inhibitors to GlyT2 that was embedded in a membrane environment. And we set up the simulations so that they would closely replicate electrophysiology experiments so that the bioactive lipids could not access the intracellular side of GlyT2 and could only bind to the extracellular membrane and GlyT2. So what we found is that in all of these cases, every simulation system we set up, we had a number of bioactive lipids in there, and they would form micelles in the solution. Some of them would bind to the lipid head groups and partition into the membrane. But in each case, we had one bioactive lipid that would bind to GlyT2 from the solution and bind to this site here. And from that, we proposed that there was a novel binding site, an allosteric binding site that was different to the substrate binding site for GlyT2, for these bioactive lipids on GlyT2. And so we compared this to the mutagenesis work that had been done in Rob Vandenberg's group. And Rob did a series of mutations in GlyT2. And these ones in red here, he found that they impacted the binding of or the inhibition of GlyT2 in the presence of these bioactive lipid inhibitors, and the mutations in cyan had no impact. Now, when we looked at these and mapped them onto the structural scaffold of GlyT2, we found that these all were situated around the area where the bioactive lipid, which is in pink here, bound to it. And from this, we could really kind of clearly point out that this site corresponded to an extracellular allosteric site out here, and it was quite different from the substrate binding site and the vestibular allosteric site of GlyT2. So we found a novel allosteric site that binds bioactive lipid inhibitors, and these lipid inhibitors spontaneously bind to this site in simulation. So this is good because at the moment we're at a point where we're starting to replicate the electrophysiology experiments and find a reason for, or a rationale for the inhibition of GlyT2 by these lipids. So we could use these and the combination of these lipids here to start to put together a structure-activity relationship for these different lipids. And so Tristan Rawling is a very talented medicinal chemist and can do all sorts of magic that I can't do. And what he could do was he could synthesize these lipids where he varied the stereochemistry of the lipid head group. So he either did L or D head group stereochemistry. He could also vary the length of the acyl chain on the lipid and the position of the double bond in each of the lipids. And from here, we could start to put together a structure-activity relationship. So we found that the head group stereochemistry, we had lysine was a more potent inhibitor than tryptophan, and L-lysine was more potent than D-lysine. Likewise, L-tryptophan was more potent than D-tryptophan. And you can see here from examining the binding of L and D tryptophan to GlyT2, and I've just superimposed the poses from two different simulations here, that the L-tryptophan, the acyl tail of that bioactive lipid goes straight down into this binding cleft that we have named the lipid allosteric site. But D-tryptophan, the different conformation of stereochemistry conformation of the head group, means that the tail does not sit straight down into this lipid binding site and is off to an angle. And we can see the effect here on the IC50s of each bioactive lipid. The next thing we wanted to do is have a look at what happens with tail length. And we found that the C18 tail was more effective for inhibition than C16 or C14. Reflected again in the IC50s. And then we could also look at the effect of double bond position on this, where we have C18 omega-3 is more effective than C18 omega-9 and other bioactive lipids. So we can now start to tease apart the interactions. We know we need a long tail. We know we need an omega-3 position for the double bond. And we know we need the L versus D stereochemistry of the head group. So the really interesting thing happened when a postdoc who was formerly in my group, Katie Wilson, went back and had a look at these simulations when we were doing all of this and said, hey, there's something weird that's going on. Whenever we do these simulations, we have cholesterol coming in from the intracellular leaflet of the membrane, and it spontaneously binds at a point on GlyT2. And then it does something really weird. It flips orientations so that the hydroxyl head group of cholesterol is no longer facing towards the membrane and instead starts to face towards the middle of the membrane and then interacts with the bioactive lipid tail. So we found this was really cool, and we wanted to investigate this a little bit more because it's starting to show us that perhaps cholesterol, the binding of these bioactive lipids to GlyT2 is a cholesterol-dependent mechanism in here. So just to back up a little bit, GlyT2 is an SLC6 transporter, and as I said before, it has a range of conformations where it has an outward open conformation where it's open to the extracellular environment, and an inward open conformation where it's open to the intracellular environment. Now, from structures of other SLC6 transporters, we know that there is a lot of cholesterol and other crystallographic solutions binding to specific places on these structures. And in particular, there's 6 different cholesterol binding sites when you look across the range of SLC6 crystal structures. And one is known as the COL1 binding site, and it's over around the back of the transporter here, down on this side. And this is actually where our cholesterol from the membrane is recruited to and binds to on GlyT2. So what we find here is we have the COL1 cholesterol binding site on GlyT2, and this is immediately below the lipid allosteric site that we proposed from these simulations. So what we have is a mechanism where bioactive lipids insert into the lipid allosteric binding site, and then cholesterol is recruited from the intracellular leaflet of the membrane and undergoes this weird reorientation within the membrane to bind to GlyT2 and stabilize the lipid binding. So this was really cool. And what we wanted to know was, firstly, is this real? Does cholesterol really influence GlyT2 transport? So we went back and talked to Rob Vandenberg about this, and we came up with this plan. So what Rob's group did was they tried to do cholesterol depletion, or they did cholesterol depletion of the xenopus oocytes. And so they ran controls where there was no cyclodextrin present, and then they depleted the cholesterol with 15 millimolar cyclodextrin added to the solution surrounding the xenopus oocyte. And then they administered the bioactive lipid inhibitors through electrophysiology, and they found that in the presence of 15 millimolar cyclodextrin, you had this depletion in current, indicating that there was a cholesterol-dependent mechanism at play for the function of these bioactive lipids. So the bioactive lipids are dependent on the presence of cholesterol. That was the initial thought with this. And so Rob's group went on and did other work on this and looked at what happens to inhibitor potency and also recovery. So you can see that in each case with oleol L-leucine and oleol L-lysine as well, so 2 of our most potent bioactive lipids, that cholesterol depletion alters the potency of the inhibitors and alters the recovery from GlyT2 from the activity of the action of the inhibitors to differing degrees. So we do have a cholesterol-dependent mechanism at play, or according to our cyclodextrin depletion experiments. So we wanted to go on and see what happens if we mutated different residues within what we propose is the bioactive lipid binding site and the cholesterol-1 binding site of GlyT2. And in particular, we found this residue here, tyrosine 430, and we have 2 different effects with tyrosine 430. If it's mutated to a leucine, then we have an increase in the recovery of GlyT2 when the bioactive lipids are administered. And in simulations, we have binding of cholesterol in this kind of position here with interacting with tyrosine 430 just here. Now, if this tyrosine 430 is mutated to a phenylalanine, we get a decrease in recovery, and we have a differential binding interaction of cholesterol in MD simulations. So this is telling us that something different is going on, and tyrosine 430 is a key interaction that's maintained here. And what we can actually see here from MD simulations is tyrosine 430 is part of a hydrogen bonding network that forms with the hydroxyl group of cholesterol when cholesterol is sitting in the COL1 binding site. And this hydrogen bonding network is present for over 92% of the simulation when cholesterol is present. So our mutation of this residue to either a phenylalanine or a leucine alters and perturbs the hydrogen bonding network, which has variable effects on cholesterol, the presence of cholesterol binding to this binding site. You can see up here an inhibitor bound to the lipid allosteric site and how close that actually is to the cholesterol that's bound in the at the COL1 site. So what we found is basically that the tail length and the double bond position of the bioactive lipid inhibitor is critical for the interaction with cholesterol. And so what we want to see, or what we saw in MD simulations, is this hydrogen bonding interaction with cholesterol, and then an interaction where the bioactive lipid inhibitor tail came down and formed a close contact with the styrene ring of cholesterol. One of the next things we wanted to know within this process is whether the current GlyT2 inhibitors were cholesterol dependent. Now, there is one other inhibitor of GlyT2 that's kind of the gold standard for inhibitors. It's a compound named ORG-25543, and unfortunately, it's an irreversible inhibitor of GlyT2. And one of the things we were doing was examining whether or not ORG would bind in the substrate binding site, which is close to the vestibular allosteric site, which sits just up here. This site, the combination of the two sites is sufficient for ORG to occupy. It can't just occupy the substrate binding site. And we also looked at whether or not it would bind the lipid allosteric site. So we wanted to do simulations of both of these to see if we could tease apart which binding site ORG actually bound to and whether or not it was a cholesterol-dependent mechanism. So Rob's group did a whole lot of mutations in the vestibular allosteric site, the substrate binding site, and also the lipid allosteric site. And the mutagenesis results indicated that ORG would bind to the lipid allosteric site. We also used the same protocol in MD simulations to look at, we docked ORG at either the substrate binding site or in the lipid allosteric site. And then we did an energy decomposition, and we found that the free energy of binding was far more favorable in the lipid allosteric site than in the substrate and vestibular allosteric site. We also noted that the magnitude of this energy is indicative of an irreversible binder for this process. So from this, we believe that ORG binds to the lipid allosteric site rather than the substrate allosteric site, and acts as an allosteric inhibitor rather than a competitive inhibitor for GlyT2. Now, when we looked at cholesterol depletion experiments, but when Rob's group looked at cholesterol depletion experiments, they found that cholesterol mediates the reversibility of ORG, but not the potency of ORG. So this is an interesting result, and we're still trying to work out exactly what happens here. So we can see that ORG binds in the lipid allosteric site, and we have several molecules of cholesterol that interact with GlyT2, and then one of those goes to interact with the COL1 site. But, you know, this interaction, the presence or absence of cholesterol does not affect the potency of ORG. It's always a very potent inhibitor. So this brings us to another question with all of this. And I guess this is one of the reasons why I'm here at this seminar series, is what happens to the membrane. So we've got these structures and simulations of membrane proteins. But one of the things, in a lot of ways to look at these experimentally, we're just seeing the actual structure of the protein. We might see one or two lipid molecules or detergent molecules bound at particular places on the transporter. But what actually happens in the membrane? And this is one of the big questions that my group is really interested in. Now, if we think back to neuropathic pain and chronic pain, if we have inflammatory pain, we have increases in reactive oxygen species in the intracellular environment. And this increase in reactive oxygen species is an inflammatory process that then alters the membrane lipid composition, because reactive oxygen species can react with cholesterol to oxidize it. Now, there are several different ways that cholesterol can be oxidized, but if we have a think about, or if we just look at the research that's out there on things like Parkinson's disease and late-stage Alzheimer's disease, what they find is at up to 20 molar percent of the lipids in the membrane are oxysterols. So this is just a bit of an infographic about what would be in a normal MD simulation. We would have something like a POPC membrane, and we'd incorporate, say, 20% of cholesterol in there to replicate what's happening in an oocyte. A real neuronal membrane has many hundreds of different lipid species, and we've just depicted them by head group species here. And a neuronal membrane is actually up to 44% cholesterol. Now, if we think about a late stage Parkinson's or Alzheimer's membrane, where we've got up to 20 molar percent of the membrane being oxysterols, that means that almost half of the cholesterol in these neuronal membranes are oxysterols. And you would think that this would have a big impact on the properties and the behavior of the membrane itself. And in previous work that I did way back at the dawn of time, we found that in a lot of membrane proteins, such as this one here, P-glycoprotein, it's a multi-drug transporter, that the composition of the membrane actually dramatically alters the conformation of the protein. And in a membrane that does not contain cholesterol at all, P-glycoprotein changes conformation and actually has a dissociation of these nucleotide binding domains from the transmembrane domains, meaning that the transporter is non-functional. Now, when you change the membrane composition to include just 10% cholesterol in this particular case, you come up with a stable conformation of this transporter so that you can start to see what's going on. So this is a really important thing when we're looking at transporter function. Other things that's important to remember about the membrane is that cholesterol concentration modulates things like the membrane thickness, which then influences the area per lipid, so that a membrane that has a higher concentration of cholesterol has a thicker membrane and it's more ordered. It also changes properties like cholesterol flip-flop across the membrane and the permeation of small molecules into the membrane. So having known this, it won't surprise you to find out that when you have a membrane that has more cholesterol, alterations in cholesterol, the presence of cholesterol within the membrane alters the accumulation and partitioning of drugs into the membrane, such as doxorubicin, paclitaxel, and nicardipine, which are two chemotherapies and a sodium channel blocker. It also changes the accumulation of morphine within the membrane, which is of more relevance to this particular study. So the presence of cholesterol and the composition of the membrane really do impact what's going on with these transporters. So with that in mind, we asked ourselves, what's a good computational approximation for a synaptic membrane? We know that we have an increase in oxysterol accumulation within the membrane. And there's a number of different oxysterols. So one of our questions was, how many different forms of oxysterols are there, and how can we start to represent these? So there's a number of enzymes that are involved in the enzymatic accumulation of oxysterols, and these are usually for trafficking out of the cell. And then we have non-enzymatic induction of oxidation of sterols through reactive oxygen species, and there's a number of different ways that this can happen. These are the main products here, and basically we can group them as either a ring oxidation approach up here, where we have an oxysterol attached to one of the steering rings of cholesterol, or a tail oxidation down here, where we have an oxidation within the cholesterol tail itself. And we can use coarse-grained molecular dynamics simulations to map either of these oxidation sites of cholesterol into a coarse-grained approach where we have 8 supramolecular beads, and we have an oxidation site either within the ring system or in the tail system of cholesterol. And then we could map this into a coarse-grained representation of the neuronal membrane to see what happened and how the position of the site of oxidation in cholesterol affected the properties of the membrane. So one thing we found very rapidly was that the site of cholesterol oxidation does impact membrane properties. And if you can just see this red curve here, this is the relative density of cholesterol in the membrane, and you can see that cholesterol sits just below the phospholipid head groups of the phospholipids, and it's kind of distributed throughout each leaflet of the bilayer. When you have an oxidation site in purple here of cholesterol, we tend to find that this molecule is more polar, and it sits closer to the phospholipid head groups and is more hydrated than a normal cholesterol molecule. In contrast, if you've got a tail oxidized cholesterol, where the normal hydroxyl group of cholesterol is in red, and the tail oxidized hydroxyl is in purple here, then you've got a very different situation. And you've got an accumulation of cholesterol in the membrane, and there's always a polar group in the middle of that membrane. So we're going to have an accumulation of polar groups in the middle of the membrane, and we actually see this bobbing behavior of cholesterol where it rapidly bobs between one leaflet and the other rather than flip-flopping in the normal process. So this was really interesting, and it started to explain what's happening with some of these interactions. We wanted to see what happens to the bioactive lipids in the presence of these oxysterols. So in the case of cholesterol, we saw that it adopted this weird position where the hydroxyl group of cholesterol was actually facing the middle of the membrane and interacting with the tail of the bioactive lipid. In the case of the ring oxidized cholesterol, we have cholesterol sitting flat in the COL1 binding site and interacting with the lipid head groups that you can see in grey here. So it adopts a very different pose to what normal cholesterol does within the membrane or within the COL1 side of Glycinergic. And when we had a look at this, the behavior of this, and counted this over a number of simulations, we could look at the average time that cholesterol or ring oxidized cholesterol interacted with the COL1 site of Glycinergic. And we find this rapid exchange that you can see in the video here, where each of the cholesterols and ring oxidized cholesterols are colored differently. Just in case you didn't get that, I'll go back. And you can see the rapid exchange between these oxysterols, and this happens throughout the simulation. And we have on average around less than 2 microseconds per interaction time of the sterol, and it's always constantly being replaced in that COL1 binding site. When we then went to look at tail oxidized cholesterol, we saw a very different thing happening. So here you can see tail oxidized cholesterol in the extracellular leaflet of the membrane. And you can see it really doesn't want to bind at the binding site. However, there's one interaction, and it is a very persistent interaction, which was maintained for over 10 microseconds in the simulation, where tail oxidized cholesterol comes in and binds at the COL1 binding site. And then it causes the bioactive lipid to leave the lipid allosteric site. So this was a really different interaction. So in terms of tail oxidized cholesterol, what we noticed was there's this residue here, a lysine 197, that interacts with the hydroxyl group in the tail oxidized cholesterol. And we also have tyrosine 430 up here interacting with the hydroxyl polar head group of this tail oxysterol. And this stabilizes the binding of this oxysterol in the COL1 site. Interestingly, when we go back to have a look at ring oxidized cholesterol, we find that when ring oxidized cholesterol is in the COL1 site, we have very persistent and long-lasting binding of a bioactive lipid within the lipid allosteric site. But in contrast, when we look at what happens with tail oxidized cholesterol, we can see that that bioactive lipid dissociates very readily from the site. So what this is telling us is it depends to get a therapeutic that actually selectively inhibits a particular type of pain, you need to know what's in this membrane environment and see whether or not you've got a ring oxysterol or a tail oxysterol. So the species of the lipid within the membrane is going to impact the effectiveness of the analgesic in the site. So in summary, what we can say is the current generation of GlyT2 inhibitors and all our bioactive lipids that are lead compounds are cholesterol dependent and non-competitive compounds that do not compete with the binding of the glycine substrate to GlyT2. We can also see from the oxysterols that studying a transporter and a pharmaceutical or pharmacological lead compound in isolation is not enough, that we really need to consider the complexity of the membrane and what's happening in that particular disease mechanism to be able to work out how to tease apart these interactions and see what is going to work for a particular therapeutic for chronic pain in this particular case. And the different membrane lipids have different contributions depending on their disease-specific contexts. And one of the questions we're now asking is, can we look at oxysterol accumulation patterns to target specific conditions or to exploit these interactions for new conjoint therapies? So with that, I'd like to thank the people in my group who did all the work. And this, as I said, this was a very long-term project. It started out with Dr. Alexandra Schumann-Gillett, and then we had Katie Wilson, who is now has a faculty position at Memorial University of Newfoundland. And it was continued on by Heather Aitken, Ada Quinn, and Patrick Sutton. From University of Sydney, Rob Vandenberg has been a really significant collaborator of mine over the last 10 years. And people in his group who are involved in this project, Shannon Moston, who's now gone on to a postdoctoral position elsewhere, Zach Frangos and Ryan Cantwell Chater, and also Tristan Rawling and Chris Sanofi from New York. So with that, I'd like to thank you for your attention. Also to the people who provided the supercomputer time for us, and the NHNMRC and ARC for their funding.
Okay, thank you very much, Megan. That was a really engaging and interesting talk. We've now got 10-15 minutes for questions. So if anyone has questions, either put your hand up through Zoom or you can ask it through the chat and I can pass the question along. I might ask a quick question first, if I can't, if no one else has one. A bit more of a general question, but what do you mean by coarse-grained molecular
Oh, okay.
dynamics?
Yeah, okay. So there's a few different approaches here. So I'll jump back to a different slide. So when you have atomistic molecular dynamics, you're representing every atom in the system as its own explicit um, atom, bead, whatever you want to call it. So in coarse-grained molecular dynamics, what you do is for every 4 non-hydrogen atoms, so a group of 4 carbons or carbon, oxygen, whatever the non-hydrogen atoms are, and their related hydrogens, you represent this as one, I guess, superatom, so a coarse-grained bead. So then all the parameters are able to be mapped down to that. The advantage of this is it decreases the number of atoms that you're trying to simulate. So it allows you to simulate for a lot longer. And it also smooths the energy surface so that you can look at diffusive processes happening quite quickly. And that might not be something you can do in the time scale of an atomistic simulation.
All right, thanks for that. We've got a question from Alex.
Hi, Alex.
Hi, Megan. Great talk. Thank you. Really interesting stuff.
No worries.
Um, I had a question actually on this slide and
Yep.
about the oxidized sterols and cholesterols in general. So um, how does this happen in vivo? It's like how common are oxidized cholesterol molecules in the synapse? And like, is this measured or, what are the processes that contribute to that basically in a broad sense?
Yeah, it's a bit complicated because usually ethics experiments don't let you look at live synapses, you know, of people who are not sick. So most of the data that's been gathered on this has either been cell lines or mouse models of inflammatory processes or post-mortem results. So we know that in very late stage neurodegenerative diseases that there is a significant accumulation of oxysterols. Um, Oxidation of sterols and lipids is a very common process. Basically, the double bond in any phospholipid can easily be converted to basically oxidized bioreactive oxygen species within the cell. And once this starts to happen, then it sets up a reactive oxygen reactive cascade where you have accumulation of reactive phospholipids and cholesterols, oxysterols. So this can happen. And it has been shown to be important in other conditions as well, other diseases. For example, muscle myopathies and so forth. So these things do happen. How common it is and how much oxysterols there are in kind of a normal person's brain, I'm not really sure. I don't think that there's any precise data out of that. But, the half-life of cholesterol in the brain, in an adult brain, is something like 5 years. So it's actually a really, really long half-life. So you're not clearing a lot of cholesterol readily. You don't have a huge turnover of cholesterol in an adult brain. So once you do start to damage it, you can imagine that it would kind of,
Yep.
mechanism where it sticks around.
Yeah, really interesting. Well, I mean, we're starting to look at cholesterol in plain and lipid bilayers and incorporating membrane proteins in there. And so we're just playing around the percentage of cholesterol at the moment, but we're now to think about the oxidation state as well. So it's food for thought. Thank you, Megan.
No worries.
We've got another question from Gio.
Hi, Jill.
Yes, Megan, can you hear me?
Yes, I can.
Yeah, wonderful talk. Thank you very much. And I have, well, two related questions
Yeah.
around the cholesterol composition. And have you ever changed or increased the cholesterol like a concentration in your simulation, or you use a fixed amount of that then just change the like for here, change the tau or ring oxidation position?
Yeah, we have. We've got a paper. It's by Katie Wilson as well as the first author. And it was published in, can't remember whether it was 2020 or 2021, where we worked up from a simplified membrane that contained POPC and 20% cholesterol to the composition of a neuronal membrane by altering the head group, lipid head group species, the cholesterol concentration, and also the lipid tail species in a systematic manner. So we find that when you change the composition of cholesterol in a simple membrane, and lots of people have done that, then you get a big impact on the properties of the membrane, the thickness of the membrane, and also the area per lipid. And it tends to make a membrane go more into a gel phase than in the liquid phase that you associate with the normal cell membrane.
Yeah. And then another follow-up question is that with respect to the lipid species in your simulation, is that you just use POPC to form the bilayer, or you also include other kind of lipid?
So it depends. So in the earlier work that I presented in the first half of the talk, we used a membrane that was 80% POPC and 20% cholesterol. And this was before supercomputer power was enough for us to do really big membranes and really complex membranes. So one thing that Ada is doing throughout her PhD is looking at Glycine-2 in these complex membranes. We've done a little bit of this in the past, but now we're trying to work out what happens if we look at oxidized species and so forth in these membranes as well. So in these membranes here, we've actually got over 50 lipid species when you take into account the head group diversity of the phospholipids, the tail diversity of the phospholipids, and then cholesterol and oxysterols.
Oh, cool. Yeah, it's very interesting combination, especially seeing different like oxidation side of the cholesterol can really change the binding of the or NAM, probably or allosteric inhibitor. That's very interesting one. Thank you very much.
No worries.
We have time for one more question if anyone had one. If not, I might ask one one last one. Yeah, so I know you more focused on the molecular dynamics rather than the electrophysiology, but I was wondering if you were able to play around with any membrane dynamics or compositions when it came to the electrophysiology stuff.
Yeah, so I mean, the problem with that is that the electrophysiology is best done in xenopus oocytes. They're a nice big cell. And so because of that, Rob's been able to do the cholesterol depletion experiments. I know that he has attempted to incorporate the oxysterols, but there's also, experimental limitations like things crashing out of solution and that kind of thing. So it's not as easy to do that. One of the things that I find really intriguing is that if you have a look at, say, xenopus oocytes compared to hex cells compared to, any other cell type, all have very different compositions. So the composition of the plasma membrane of a cell is intrinsically related to the cell type. And so it really depends where your cell is from, what media it's growing on, and all of those other kind of biochemical and environmental factors as to what the composition of the lipid membrane will be. I think it's a huge area that membrane proteins, it's huge scope for membrane proteins there.
Yeah, lots of lots of stuff to
Lots to think
account for,
about.
definitely. Well, thank you very much for your time. It was, as everyone said, a great talk. So with that, I think we'll wrap up. I'd just like to remind everyone of our next seminar date, which will actually be October 1st at 4 p.m. We've got Dr. Basil Graeber speaking to us, so it'll be great to see you all then. And thank you once again, Megan, for your time.
No worries. Thanks a lot for inviting me, and I hope to see some of you at Biomolecular Horizons. Okay, see ya.
Definitely.